Hey, Pharma subscribers! Today, we're diving into a really important topic for anyone in the pharma industry, the ICH Q2R2 guidelines for analytical validation. This updated guideline is super crucial for ensuring the reliability and accuracy of our analytical methods. It covers everything from validating specificity, accuracy, and precision, to things like linearity and range. Understanding these guidelines is key to making sure our data is robust and meets all the regulatory standards. It helps us ensure the quality and safety of pharmaceutical products. So, whether you're a lab technician, a research scientist, or just interested in the pharma industry, getting to grips with ICH Q2R2 is essential. Let's keep learning and raising the standards in pharmaceutical analysis together. Don't forget to like this video, subscribe to Pharma and hit that notification bell so you don't miss our future content. All right, let's talk about precision in the context of ICH Q2R2. Precision refers to the closeness of agreement between a series of measurements obtained from multiple samplings of the same homogeneous sample under the prescribed conditions. There are a few different types of precision we consider: repeatability, intermediate precision, and reproducibility. Repeatability is the precision under the same operating conditions over a short period of time, using the same analyst and equipment. Intermediate precision considers variations in different analysts, equipment, or different days. Reproducibility is the precision across different laboratories. For the ICH guidelines, you'll need to demonstrate precision through tests like repeat analyses of a homogeneous sample and calculating the relative standard deviation. The specific requirements will depend on the type of analytical method you're validating and the intended use. It's important to follow the guidelines closely to ensure the reliability of your results and meet regulatory expectations. Building on precision, let's talk about accuracy. According to the ICH Q2R2 guidelines, accuracy refers to the closeness of agreement between the value which is accepted as a conventional true value and the result obtained by the analytical method. In other words, it measures how close your test results are to the actual value. To assess accuracy, you typically perform tests by applying the analytical procedure to samples with known concentrations, such as reference standards or placebos spiked with the analyte. The acceptance criteria for accuracy are usually expressed as the recovery rate, which is the percentage of the known amount that is detected by the method. High accuracy is crucial for ensuring the reliability of your analytical results, as it indicates that the method is providing values that are close to the actual concentration of the analyte. It's a fundamental parameter for validating analytical methods and ensuring the quality of pharmaceutical products. Sure, let's discuss specificity as outlined in the ICH Q2R2 guidelines. Specificity refers to the ability of an analytical method to unequivocally assess the analyte in the presence of components that may be expected to be present. This is crucial to ensure that the method accurately measures the target analyte without interference from other components such as impurities, degradants, or excipients in the sample matrix. According to the United States Pharmacopoeia, USP Chapter 1225, Validation of Compendial Procedures, specificity is a critical parameter. The ISO 17025 standard, which covers the competence of testing and calibration laboratories, also emphasizes the importance of specificity for ensuring the reliability of analytical testing. To demonstrate, let's move on to linearity. According to the ICH Q2R2 guidelines, linearity describes the ability of an analytical procedure to obtain test results that are directly proportional to the concentration of the analyte within a given range. Essentially, it measures how well the method's response follows a straight line across a range of concentrations. To evaluate linearity, you typically prepare a series of solutions of the analyte at different concentrations and analyze them using the method. The data are then plotted, often as the response of the method versus the concentration, and analyzed using linear regression. The correlation coefficient, or coefficient of determination, is used to assess the linearity. High linearity indicates that the method provides consistent results across a range of concentrations, which is essential for accurate measurements. Both the USP and the ISO 17025 guidelines emphasize the importance of linearity in analytical validation. The USP provides specific requirements for assessing linearity, depending on the type of method and its intended use, while ISO 17025 focuses on the overall validation of methods within laboratories, including the assessment of linearity. Ensuring linearity is crucial for obtaining reliable and accurate quantitative results, particularly in pharmaceutical analysis. The detection limit is a crucial parameter in analytical method validation. It represents the lowest concentration or amount of an analyte that can be reliably detected by the method, though not necessarily accurately quantified. It's often determined by analyzing samples containing the analyte at very low concentrations or by using the signal-to-noise ratio of the method. A method with a low detection limit is considered more sensitive, as it can detect smaller amounts of the analyte. This is particularly important in applications where the concentration of the analyte is expected to be very low. So, like I was saying, the quantitation limit, or LOQ, is the lowest concentration of an analyte that can be reliably quantified with acceptable accuracy and precision. It's super important for methods where you need to measure small amounts of stuff. ICH-Q2R2 gives you different ways to determine the LOQ, just like for the detection limit. Alright, so moving on to robustness, think of it as the method's reliability even when there are small changes in the conditions. It's like checking if the method still works well if the temperature fluctuates a bit, the pH is slightly different, or the flow rate changes. You deliberately vary these parameters within a certain range and see if the results are still consistent. This helps ensure that your method is reliable even under small operating conditions, where there might be minor variations. It's about proving the method isn't too sensitive to small changes and can still provide accurate results reliably. This is especially important for methods that will be used in different labs or by different analysts, ensuring consistent results across different environments. Reporting and documentation are absolutely critical in method validation. It's not enough to just perform the experiments. You need to keep a detailed record of everything. This includes the validation protocols, which describe exactly what you plan to carry out the validation studies. Then you need to document all the raw data and results obtained from the experiments. Finally, you summarize your findings and conclusions in a comprehensive report. This documentation is crucial for several reasons. It provides a clear record of the validation process, allowing for traceability and transparency. It's also essential for regulatory compliance, as authorities often require detailed documentation of method validation. Furthermore, it allows for future reference and troubleshooting if any issues arise with the method later on. Think of it as the scientific story of your method, providing all the necessary details to understand its development and performance. Following linearity, let's discuss range. In the context of the ICH-Q2R2 guidelines, the range of an analytical procedure is the interval between the upper and lower concentration levels of the analyte, for which the analytical procedure has been demonstrated to have the required precision, accuracy, and linearity. It basically defines the concentration range where your method is reliable. To determine the range, you need to validate the precision, accuracy, and linearity within the specified limits. The USP and ISO 17025 guidelines provide specific criteria for establishing the range, depending on the type of analysis, such as for assays, impurities, or dissolution testing. It's crucial to ensure that the method is valid across the entire range where it will be used, to ensure the reliability and accuracy of the results for different sample concentrations. Establishing the appropriate range is vital for demonstrating the suitability of the analytical method for its intended use. Okay, let's talk about the overall method validation process. It's basically a structured approach to make sure your method is fit for its intended purpose. It involves careful planning, where you define the validation experiments and acceptance criteria. Then, you execute the studies according to the plan, collecting the data to assess all the relevant characteristics like accuracy, precision, and robustness. Finally, you document everything thoroughly: the plan, the results, and your conclusions. It's crucial that the validation process is tailored to the specific stage of your product. For example, the requirements might be different for a new method being developed for research compared to a method used for routine quality control in manufacturing. You need to ensure the validation is appropriate for the intended use of the method. It's like building a strong foundation for your method, ensuring its reliability and validity throughout its life cycle.